Okay, so we were talking about like what exactly is prosite syntax pattern. Okay, what are the rules that are needed to be followed when you formulate any short motif in prosite syntax pattern. Okay, and according to this prosite syntax pattern, the amino acid residues in that particular motif are separated by a hyphen, or you can say dash. Okay, and ambiguities are represented either in square bracket or in curly bracket or in round bracket. Okay, so these ambiguities would mean different as per the bracket that it has been placed in, right? And there are also rules for determining if the sequence is present on n terminus or if the sequence is present on c terminus, and so on and so forth. We have kind of discussed it in our previous section. Okay, so now let us talk about what are the limitations of using this prosite pattern. Okay, so now first and foremost, it is very evident that this prosite pattern would be an effective way to describe a motif only when the domains, or the sequences that are being considered for creating that prosite pattern are functionally very conserved, or they are quite well conserved uh, in the sequences as such. Okay, so now the thing is that very few such domains are known that are found to be conserved throughout evolution in a very perfect way. Okay, well, say for example, the domains or the proteins which are present in globin family. Okay, so proteins which are present in globin family might include your hemoglobin, it might include your myoglobin, androglobin, neuroglobin, which are present in vertebrates. In plants, you can have leg hemoglobin. That means this globin family is conserved throughout evolution. Okay, another class of family that you might know is immunoglobulin, that is the protein containing the Ig domain. We know that there are at least 765 members so far identified in this immunoglobulin family. Okay, there are certain proteins which are having SH2 or SH3 domains. Okay, these are the proteins with SRC homology domains. Okay, and these protein might act as adapter in the signaling network. Okay, so the thing is that there are very few domains which you can say that they are actually conserved throughout evolution. Okay, and hence the prosite pattern or prosite syntax or whatever motif recognition code that you might develop, it might appear very well in such highly conserved domain. Okay, so now it is entirely possible that in due course of evolution, the domains or the sequences which are important for performing certain functionalities might diverge with time. Okay, they might accumulate mutation. Okay, and hence when the sequences are quite well diverged across different genera, different species, then it might become difficult to identify distant homologue. Okay, so in such case, your prosite pattern would be ambiguous. It would contain lot of variation that would be very difficult to account for. Okay, so in such case, the prosite pattern are ineffective for deducing relationship among the given sequences as such. Okay, so you might end up having certain pattern that is quite well ambiguous, and you might miss up some important site if they are already there, or you might end up identifying something which is not there. Okay, so these are the problem that would appear when you consider sequences that are distantly related to each other across genera or even division or classes. Okay, so identifying distance homologue with prosite pattern becomes a difficult task. Okay, so now in such a case, in order to identify such distantly related protein sequences and the domains and the functionalities that is involved in such domains, we generally use profiles instead of patterns, right? So let us talk about prosite profiles first. Okay, so now these profiles, well, not only as a signature from prosite database, but there are also a few other databases that also contribute to, you know, to provide the signature of profiles like HAMAP and PRODOM, which we are going to talk in a little while. Okay, so in general, profiles are based on weight matrices rather than the order of amino acid residues that might occur in the protein sequence. Okay, so now since we are trying to classify the protein sequences on the basis of weight matrices in or you can say in place of the actual pattern of or actual motif of these sequences, it would allow you to detect some poorly conserved domains or families which are well scattered across numerous genera or numerous species. Okay, so in this way, uh, patterns characterize the protein domains over the entire length, not just most conserved part of it. That was in case of patterns as such. Okay, तो यहाँ पर profiles रहना चाहिए. Profiles, sorry, just pardon this. Here should be profiles. Okay, so profiles characterize the protein domains over the entire length. On the other hand, your pattern were able to characterize only small portion of the domain. Okay, only small short conserved motif of your domain that might act as providing certain function. Okay, so here is the basic difference between profiles and patterns. Okay, just pardon here. Here should be profiles. Okay, so I need to correct it out. Okay, so now the question might arise ki, either now you have a chance to do functional analysis with pattern, or you can do profile. Then where to rely? Okay, when to use what? When to use profile and when to use pattern? Okay, so what is needed to be used remains very context specific. Okay, so both the descriptors that we have talked so far, that is, I'm talking about your patterns and profiles. So both of these descriptors have complementary qualities, right? Well, now if your objective is to identify a protein's function, such as if it is involved in say some enzymatic activities, patterns would be a more suitable means to do that. Okay, why? Because patterns are confined to small region with high sequence similarity, and that is the exact thing when you are looking for any proteins enzymatic activities. You would be looking for active sites, right? So in such a way, if you want to identify protein functions such as enzymatic activities, you might be interested in focusing on say some small region of your protein, small motif, small local region of your protein, and in such case, relying on patterns would be more suitable. Okay, now, on the other hand, if your objective of doing functional analysis is to predict protein structural properties, then profiles can be more suitable. Why? Because structural properties, are influenced by domains, their organization, and their, crosstalk with each other, right? So, in this way, profiles cover complete domains and are not just confined to small regions with high sequence similarity. And if this is the objective of your functional analysis, then uh, relying on profiles might be a better idea. Okay, so when to do what would be entirely context specific, and you would have to decide what descriptor you are interested in. Okay, so this is all about your patterns and profiles, right? Difference between patterns and profiles and when to use what, basically, right? So now let us talk about how do we construct the profile, right? In our previous lecture, we have talked about how do we construct a pattern, prosite pattern. Now we would talk about how do we construct the profile, or you can say the profile for the given sequences as such. Okay, so now there is a very classical paper that was published back in 1990s in the journal called as Methods in Enzymology. It was a pioneering work of this Gripskov, who designed the very initial algorithm for classifying the proteins into families using the concept of profiles. Okay, so now the standard automatic procedure that was implemented in construction of profiles was in a specific program called as pf make in a package called as pf tools. तो ये जो प्रोफाइल कंस्ट्रक्शन का काम आपका जो प्रोसाइट रिसोर्स है उसके ऊपर जब प्रोफाइल पहली बार कंस्ट्रक्ट किए गए थे, दैट वेर कंस्ट्रक्टेड यूजिंग दिस क्लासिक Gripsau's original method. And in order to generate such profiles, people use the package called as pf tools. Or this being a package, there were different components that were present in this package, and in order to construct a profile, a program called as pf make was used from this pf tools package. Okay, so it is similar to ms office, right? So ms office can be called as a package which contains different programs, like you have got word, powerpoint, excel, right, publisher and whatnot. So that in similar analogy, for performing profile construction using the original Gripskov's method, pf tool package was introduced back in early 90s. Okay, and for constructing the profile, one of the tool from pf tools package called as pf make was used for generating the uh, profiles for a given set of sequences. Okay, so now I would just let you know a general procedure that was implemented for generating such profiles. Okay, so the first step that involved in construction of profile was the generation of good multiple sequence alignments of domains that were extracted from complete sequences. Okay, now you can also compare your profiles with patterns, or you can say profiles with patterns, in such a way that you can consider profiles as a global entity, right, while the patterns might be local entities. So now when we are generating profiles, that means we are, we would expect to encompass multiple domains, or you can say entire sequences when we generate profiles. Okay, rather than short region. तो इसके लिए सबसे पहले कौन सी चीज होना जरूरी है? You should be able to first construct a multiple alignment of all the domains that are present in a given sequences, okay, or domains that are somehow extracted from the given sequences. Okay, and once you have such multiple alignment, multiple sequence alignment, then what you would do, you would go on weighing the individual sequences in such multiple sequence alignment. Okay, after you weigh each and every sequence in the multiple sequence alignment, you would generate something called as frequency profiles. Okay, so from this frequency profile, you would generate unscaled profile which would be finally scaled to produce prosite pattern. Sorry, prosite profile. Okay, so these are the 5 basic steps that are involved in constructing a profile. Okay, so now this Gripskov's original method for generating the profiles make use of this PF make tool at the very last stage. That is when you create a scaled prosite pattern from unscaled prosite pattern. Yes, हाँ, profile. ठीक है. So in that case you use this PF make. But इसके पहले के भी स्टेप को बहुत सारे tools लगते हैं from this PF tools package. I am going to show them to you one after another in this particular protocol. Okay, so whatever 5 steps that I have told you, now let us go in more details of that. Okay, so you usually might begin with the protein sequences. Okay, so the first job would be to uh, to make sure how many sequence based domains are there. Okay, you can manually excise those domains from the given sequences. Okay, and then you would iteratively perform the searches. Okay, so in this way you might end up having unaligned sequences region, what we now call it as domains. Okay, so the next step would be, once you now have identified ki, okay, I have say some 500 sequences, and these 500 protein sequences might have different domains present scattered all along the sequences, right? So your first job would be to separate all domain sequences together, right? And when you have such unaligned domains in a single file, what you would do is that you would perform multiple sequence alignment using algorithms like clustral or tcoffee. Okay, so these multiple alignment, these algorithms like, or these programs like clustral or tcoffee would give you multiple sequence alignment. Okay, when you have this multiple sequence alignment, the next thing that you are supposed to do, as I have mentioned, is that you need to weigh the individual sequences in MSA, or what do we call it as multiple sequence alignment. Okay, तो ये जो weighing का काम है, it is done using the program called as PFW. Okay, and we give something called as Voronoi weights to the sequences that are present in your multiple sequence alignment. Okay, so well, let us not go into more details of how do we compute these Voronoi weights and what are the criteria of selecting the domains that are supposed to be given what kind of Voronoi weights. Well, let us not go over there. Okay, for time being, आपको क्या पता रखना है यहाँ पर कि we are supposed to weigh the individual sequences in multiple sequence alignment, and we would weigh the individual sequence by providing them something called as the Voronoi weights. Okay, and ये Voronoi weights देने वाला प्रोग्राम कौन सा है? It is called as PFW, which is an individual component of this PF tools package. Okay, so once we have weighed each and every sequence in your multiple sequence alignment, what we get is weighted multiple sequence alignment with individual Voronoi weights provided for each and every sequence. Okay, so at this point, there comes a hidden Markov model. Hidden Markov models are, using hidden Markov models, you would generate something called as frequency profile. Okay, so for profile construction method, so you might be using these substitution matrices like Blossom 45 or even PAM matrices. You might have different gap weighing parameters, and eventually using this hidden Markov model as a technique, you can prepare something called as frequency profile. Okay, so from this frequency profile, you would create unscaled profile with a program called as PF make. Okay, and once you have got this unscaled profile, over here, now you would implement the original Grips curve method, which is implemented in program called as PF scale, and that would give rise to the final ProSite profile that is now present in the ProSite profile database as such. Okay, so this is how people have created the ProSite profiles, and this methodology was so very perfect that even now, if we want to perform certain functional analysis, these scaled ProSite profiles are usually used. You can download those scaled profiles and you can compare the incoming sequence with the ProSite scaled profiles, and you might end up in classifying the proteins in a particular family or a right family, as I might say. Okay, so this is very old but very foolproof method for performing functional analysis as such. Okay, then Abhi, we have talked about ProSite that creates profile. तो ये ProSite के अलावा there are two more member databases that provide these profile signatures, that is called as HAMAP and ProDom. Okay, so these two databases also contribute something called as profiles in your Interpro consortium. Okay, so this signature is mainly used for functional annotation of protein families and functional annotation of domains. Okay, well, now let us talk about the very concept of domains. I have used the term a lot of time. Well, now when we talk about ProDom, we would be specifically talking about this domain. Okay, so now you might know that proteins are modular entities. That means a single protein sequence might contain small local region that might fold independently, and that might eventually function independently. Okay, so these modular entities, modular यानी क्या, small local entities in the given protein sequence, which has got tendency to fold independently as well as function independently, we say such entities as domains. Okay, so in such a way, there can be multiple domains present in a single protein, and each individual domain might perform its own individual function. Okay, and thus the structure of a protein might consist of single or multiple modules or units. We call it as domains. Okay, now these domains are subject to shuffle during the course of evolution. This can happen. Okay, due to genetic events, it is entirely possible that these domains can change their position. They can get shuffled. Okay, but however, if they shuffle, they get from if they get transferred from N terminus to C terminus or in the middle of the protein sequences, they usually form a structurally autonomous and might have an independent function for each and every one. Okay, so in this way, if you have a fair idea about how many domains are there and how they are arranged and where they are present among the set of sequences, it might provide you with a clue about how these protein sequences might be related to each other. When that genetic event might have happened, that might have shuffled the domain to move from one location to another location, or what kind of domain might have been evolved to perform certain function in say some protein. Okay, so well, now let us talk about this database of domains, what we call it as ProDom. Okay, so in order to create this database, whatever sequences that were present back in 1994 in SwissPro database were imported. Okay, then the first task that was done was to perform local comparison. अभी यहाँ पर blast p क्यों किया होगा? Why not entire multiple sequence alignment? ब्लास्ट पी करने का लॉजिक ये हो सकता है यहाँ पर, बिकॉज वी आर इंटरेस्टेड इन small local regions rather than aligning the entire sequences as such. Okay, so by performing this blast comparison, you might detect if there are domains that are shuffled across the sequences. Okay, so the objective was to find out local segments amongst whatever sequence that were present at that time in SwissProt. Okay, and eventually the domain boundaries were determined by the program called as domainer. Okay, एक डोमेनर नाम का एक इंडिविजुअल प्रोग्राम था which was used to identify कि where exactly one domain ends and where exactly the another domain begins. Okay, then there was another program called as mult align that was used to generate multiple sequence alignment of the identified domain. Okay, and finally the consensus sequences were constructed for all domains from this multiple sequence alignment that were carried out by mult align. Okay, so this is how things were done. Okay, now whatever domains that were identified in such a way, each and every domain was assigned a numerical identifier. Okay, by which it became possible to identify each domain independently. Okay, and this domain can now be used to search in SwissProt, ProSite, even PDB IDs can also be used to search for domains in ProDom. Okay, so whatever multiple sequence alignment that were created using this mult-align program, you know, they can also be retrieved in this MSF format. Okay, so multiple sequence alignment, you might have learned how to create multiple sequence alignment using Clustal. तो Clustal का by default extension is what? .aln. Clustal हो गया क्या तुम लोगों का पढ़ाकर? Clustal W multiple.
No sir.
हाँ?
No sir.
No, sir.
अब तक नहीं हुआ है?
No sir. We
Okay.
are doing pairwise only.
Pairwise only. Okay, so maybe ये होने के बाद में आपको मल्टीपल सीक्वेंस अलाइनमेंट जब होगा, जब फिलहाल तो इतना ही ध्यान में रखो कि when you will read about this Clustal, when we use Clustal for performing multiple sequence alignment, it would give its output in ALN format. Okay, तो ALN फॉर्मेट के अलावा देयर इज आल्सो फाइलिप फॉर्मेट जिसमें मल्टीपल सीक्वेंस अलाइनमेंट सेव किए जा सकते हैं. There is also something called as MSF format in which you can save your multiple sequence alignment. तो प्रोडॉम में जो कुछ भी डेटाबेस जो कुछ भी डोमेन्स के मल्टीपल सीक्वेंस अलाइनमेंट है, that can be downloaded in this MSF format. Okay, so of course you can download the GIF image which would highlight the conserved domains. Arrangement can be generated, that can be downloaded. Okay, so here is what the database looked like back in say 1998. Okay, given the main ProDom database entry, you used to have this link to SwissProt. If there are certain small motifs, if they are present, they can be also be mapped to this ProSite. Okay, ProSite pattern agar yahan par hoga, so it can also be mapped with the structure if it is associated in PDB. Okay, and you can also download the entire multiple sequence alignment in in an image format as shown over here. Okay, but now things have changed. We have got a new interface that looks something like this. If you go to ProDom web page now, you might see something like this. ये ऐसा नहीं दिखेगा आपको अभी. Okay, so well, now all the functionalities are still over there. You can download as I have already told you, you can download the multiple sequence alignment of all the domains in this MSF format. Now they also support downloading the multiple sequence alignment in FASTA format. Okay, you can obtain images of those multiple sequence alignment using this link. Okay, and well, you might want to explore these things in more details. Okay, so now what is the utility of this program? Why would people try to do such thing? Why would one want to, sort out different domains from multiple sequences, try to align them, and try to, document them, or try to make a database out of them? Okay, so by using this proteome database, you can resolve the domain structure of new proteins. Okay, so using data from this proteome, you might be able to answer the questions like, how many domains are there in my given protein sequence, how these domains are arranged, and eventually you can perform, or you can say, you can have an insight of how these domains are related to each other, and eventually how the sequence would be related to the group of sequences as such. Okay, so this is the overall aim of creating this proteome database. Okay, then another database that offered the signature of profiles is in form of this HAMAP. This HAMAP stand for high quality automated and manual annotation of protein. Okay, so जिस तरीके से आपने प्रोडॉम का प्रोफाइल सिग्नेचर कंसीडर किया था, वैसे ही HAMAP भी प्रोफाइल कंट्रीब्यूट करेगा. Okay, so these HAMAP profiles are, manually created by expert curators. Okay, so they identify the proteins that are part of well-conserved protein families or subfamilies. Okay, and the major focus of this HAMAP is on microbial protein, as in contrast with the previous two profile construction method we have talked about, that is ProCite and ProDOM. Okay, so now this HAMAP is based on Swiss Institute of Bioinformatics, that is present in Geneva, Switzerland. On the other hand, your ProCite and a ProDOM are based on EBI based utilities like SwissProt and UniProt KB. Okay, so this is the basic difference. So that when we use signatures from HAMAP, that means we are also considering the data that is hosted at the Swiss Institute of Bioinformatics, or SIB. Okay. Now the final concept that I want to share with you is about prints. Okay, or what we call it as fingerprints. So fingerprints ये एक signature है which is provided by this database called as prints. Okay, so this prints is a database that contributes the signature called as proteins fingerprints. Okay, so now, well, let us first try to correlate in this functional analysis where exactly these fingerprints comes into picture. Okay, so now this fingerprint is, you can say, nothing but a group of conserved motif, and this group of conserved motifs can be used to characterize a protein family, or it can be used to characterize a protein domain. Okay, so when we say that we perform functional analysis, you might consider sequences to perform functional analysis, you might consider motif to perform functional analysis, you might consider, you can say, profiles to perform functional analysis. Another way to perform functional analysis is to identify what are the fingerprints that are present in the given protein. Okay, well, now let us talk about eventually what are fingerprints, what exactly do they do. Okay, so the concept of fingerprints is, quite very, on a similar line to that of motifs. तो फर्क क्या है मैं आपको अभी थोड़ी देर बाद बताता हूँ. Okay, so what you usually do when you construct a fingerprint is that, well, say for example, I have a family of protein called as kinase. Okay, तो kinase proteins क्या करते हैं बेटा, पता है क्या आपको?
फॉस्फेट ऐड करते हैं सर.
Okay, they perform the function of adding a phosphate. ओके, फॉस्फोराइलेशन का काम करने वाले प्रोटीन्स को अपन क्या कहते हैं? वी कॉल देम एज काइनेज. तो अभी अपने को देखो फॉस्फोराइलेशन करना है, राइट? एंड यू नो दिस इज अ वेरी बिग फैमिली, right? There are different type of tyrosine kinase, right? There are you know you can have mevalonate kinase, you can have homoserine kinase. All the kinases that are involved in signaling cascades वो अलग होते हैं, right? And now in order to perform say a kinase activity, right? क्या क्या चीजें जरूरी हो सकती है, क्या क्या चीजें रहनी चाहिए हर एक काइनेज में? कैन यू जस्ट लॉजिकली थिंक कि उसमें एक ऐसा रीजन रहना चाहिए जहां पर आपका एटीपी या कोई भी न्यूक्लियोटाइड आकर अटैच होगा जो फॉस्फेट ग्रुप डोनेट करेगा. बरोबर है? इज इट क्लियर? तो सेम प्रोटीन में एक ऐसा छोटा सा रीजन रहना चाहिए जहां पर आपका आपका सबस्ट्रेट आकर अटैच होगा, राइट? जिसमें यहाँ से वेल से फॉर एग्जांपल इफ दिस इज द न्यूक्लियोटाइड बाइंडिंग रीजन, तो उसके पास में ही सबस्ट्रेट बाइंडिंग रीजन रहना चाहिए सो दैट यहाँ पर जो कुछ भी अटैच होगा अमीनो एसिड रेसिड्यू प्रेजेंट इन दिस रीजन might take off that phosphate group and attach to whatever is present over here or whatever group is attached to over here, right? Then you can have say certain another motif which is highly conserved in kinases, जैसे कि DFG loop होता है which is somehow found to be conserved throughout most of the kinase group. So all in all, you can create a kind of scenario in which in order to perform certain function by given set of protein, there are certain motifs or there are certain small local region that are necessarily present in that family of protein. Okay, in this case, in the case of kinase, we say that there must be a DFG loop, there must be a phosphate or you can say nucleotide binding loop, there must be substrate binding loop, there must be catalytic loop जो ये सारी चीजें करता है, right? So in this way, in order to characterize a kinase family, you must have all these motif or these small short regions that are present throughout these multiple kinases. Okay, so now what people have done is that after aligning all those kinase sequences, I am just taking an example of kinase, ये बाकी के protein families को भी सही यही वाला methodology implement होगा. Okay, so now once you have identified the conserved region that are required to perform certain functionality in a given set of proteins, what you do, you would excise using a protein called as SOMAP. In this way, you would now only focus on these important small regions that would collectively form a kinase entity. Okay, then what you would do is that you would iteratively scan these sequences in a database like SwissProt and PIR, just in order to make sure that you have included far vast more query coverage, or you can say कि related sequences भी आपने इसमें include किए हैं. ठीक है? So once you have this thing, what you do is that you put all these motifs into a program called as ADSP sequence analysis package. So now this ADSP sequence analysis package would convert this sequence information into something called as fingerprint. Okay, तो हर एक मोटिफ का क्या रहेगा? एक फिंगरप्रिंट रहेगा और ये फिंगरप्रिंट का कुछ स्कोर रहेगा. Okay, well, say for example, if this is the fingerprint for phosphate binding or nucleotide binding, then if it is well conserved, तो उसका स्कोर यहाँ पर ज्यादा रहेगा. If it is less conserved, तो उसका स्कोर कम रहेगा. And collectively, these four short, now you can say fingerprints would constitute something mathematically by which you can distinguish if it is a kinase protein or related to kinase protein. Okay, so in this way, if you have such mathematically derived fingerprints for a given set of proteins, that would allow a very flexible and powerful characterization of fold and function, right? तो अभी अपन sequence compare नहीं करेंगे. Now we have far more better mathematical representation of what these motifs are. Okay, and then in the incoming sequence we would test if such fingerprints are there or not. If there are such fingerprints, that means the given set of sequences can act as a kinase. Okay, it doesn't really matter if the sequences match for this motif or not. Okay, so in this way, effective functional analysis can be carried out if you have such fingerprints data. Okay, so this concept was published back in 1994, and you still have this prints database fully functional for performing such or for calculating such fingerprints data as such. Okay, so here is how we complete our Interpro consortium. These are some important databases that I have covered. Okay, so during our subsequent classes, maybe I would now share you with the assignments. Okay, and you are supposed to perform those assignments. Well, all these resources are quite self-explanatory. Okay, now what I will do is that I would upload all those assignments to all these databases that we have talked about. Okay, you are supposed to try completing those assignments and maybe ask me questions in my subsequent classes if there are. Okay, but these resources are quite very simple. I think you people will manage it. Okay, but still, if you have problem, you are most welcome to discuss it. Okay, so किसी को कुछ पूछना है बेटा?
Sir,
Yes.
so please can you upload this PPTs also with the assignment file?
Okay. That can be done. ठीक है.
Okay, sir. Thank you.
So can you explain again after the fingerprint?
Okay, this one.
Yes, sir.
How do we obtain this? Do you, well, I am not going into algorithmic detail. All I want to express is that what exactly is the concept of these fingerprints. Okay, you can say that it is just an abstract way, a mathematical way to express the sequence patterns that are present in these motifs. Okay, more conserved are the motifs, higher would be their score. And now, instead of comparing these sequences, we would end up comparing these fingerprints, which are mathematical representation of these motifs. And hence, comparing number is quite more efficient than comparing the sequences as such. You might end up getting distantly related motifs together, and it is entirely possible that you might miss those proteins if you only rely on sequences as such. Okay, so this is the basic concept behind fingerprints.